including me, 53. And let's start, okay? So I want to ask someone, very simple, because you guys are the food scientists in the future. You're going to be the food scientists. So, Kai Shen, so you guys study a lot of sugar, right? Can you tell me about lactose? When you take lactose, what happened next? Would you so in our body, what kind of metabolisms will occur when you take lactose?
The lactase will digest the lactose.
Well, that's a very convenient way to say. Well, okay, good guess. But well, you guys have to study well. I also don't know about but I do understand the bacterial system. Okay, so we look at the bacterial system when when the bacteria receive some lactose, they need sugar, right? Just like us, we need sugar to uptake and then to do a lot of things, energy, and also what? What else? We also can build our DNA, the DNA foundation. You need sugar also, right? So so the glucose, ribose, right? Everything is essential. So we need sugar to do a lot of things. We don't have sugar, we will feel very sad. Okay, some people and feel tired. Okay, so that's a metabolism. Okay, you have to think about. So right now, I want you to connect the metabolism that you learn in human metabolism you learn from your food science teacher and food science, food chemistry, etc. Okay, you know better than me this part. I don't know about this. And you link this to the gene transcription and translation. So let's say if you have, how to say, let me draw this, probably will help you. Okay, let me draw. Okay, so can you see here? In bacteria, okay, if you have lactose, right? So the bacteria will digest this lactose into glucose and galactose. So that's an enzyme is going to play the role. Okay, so this enzyme is called what? Beta galactosidase. Okay, so we'll produce this too. Okay, so the bacteria sense that, oh, there's some food. It's called lactose, but I need this to be digested into glucose and galactose so I can absorb it, right? I'm so hungry, I'm a bacteria, right? So I need to generate, how am I going to digest this lactose? So in my genes, I have a genes, okay, can do the transcription and translation to produce these enzymes. This enzymes is called beta galactosidase, okay, this guy, okay. So I will express a lot. I'm a bacteria. I'm going to express a lot of this enzyme, okay, because I can feel my surrounding full of lactose, food everywhere, lactose everywhere. So I'm going to highly express this beta galactosidase, right, to cliff the lactose into glucose and galactose. Then I can take the glucose, galactose separately, and then to run my system, and then do the DNA synthesis because I have the sugar, energy, ATP, etc. Okay, my system work well. So here, I give you a system right now. It's a system. Okay, this is an experiment. If you still remember the experiment design, the first practical or second practical. I have lactose in plate B, and this one is plate A. No lactose. No lactose. No lactose at all. And high concentration of lactose in this plate B. So obviously this is a controller, huh? Sorry about my writing. So what happened? The bacteria over here will do what? So for this, it's very easy lah, right? You have high lactose concentration. I put this bracket is concentration now. So you have high concentration of lactose. So obviously the bacteria will express a lot of what? Beta galactosidase. Beta galactosidase, okay? Express. And what about this guy? No lactose? Would they express beta galactosidase? A little bit or no? Bao Ying. Can I ask Bao Ying? Is it Bao Ying here? Would you think? What do you think, Bao Ying?
No.
No, right? Okay. Why no? Give me some
Because
logic answer.
know the substrate so the enzyme can know what.
Okay. Also, we need to keep our system. We don't waste a lot of energy to express something that we don't want. Okay. So our system is quite clever. The bacteria are also very clever because they are very tiny, right? They won't waste any energy to express those enzymes. They are not currently necessary to have it because beta galactosidase, they will, if you express and then produce the enzymes, there's no lactose. What they're going to do? So you are wasting this enzymes, the productions of this enzyme. Okay, so there is no expression on this beta galactosidase. I just write no expression on the beta galact. I put galact, okay? You just know lah, huh? What is that? Galactosidase. So this is how a very simple way to understand what is gene regulation, okay? They regulate. Gene regulation, remember, my dear, gene regulation. So you see, they express, not express is because of what? Because your system tell, okay? So logical thinking that, okay, your system, you have a system, but I haven't tell you the system yet, right? Use your logical thought, okay? Saying, telling that, okay, so no lactose, that means we're not going to express this beta galactosidase, right? Right. But how? In a molecule biologist system, how you can tell this story. So basically, that's a gene structure, okay, you need to learn. So here, okay. I'm not really good right now. Okay, here. Okay, so this is a gene called LAC-ZAC gene. That's a LAC-Y gene. And that's a LAC-A gene. Okay, so what happened here? You have a, oops. This is O. Oh yeah, 100. Yeah, I didn't do well. Okay, here. Okay. And far, far away. Okay. Okay, so here. P, lag P. So this is a lag operon. Just remember. This is a lag operon. Okay, this lag operon contain a P means promoter. You already learned, so you already know what is promoter. One is Tata box, one is what? No name, but it's in -35 region. Tata box is in -10 region. Don't tell me you don't know after I put so much effort in the tutorial question. And lag gene later lag O gene is operator. This guy. Lag O gene is called operator. And lag Z gene is called lag Z gene. Okay. So this lag Z gene, lag Y gene, okay, is involving with lactose metabolism. Lactose metabolism. Okay. Okay. So this is a gene DNA. This one is DNA. Okay. So what happened is transcription occur, right? Transcription. What happened? What happened? So what happened is what happened. This is a messenger RNA. Okay, 5' to 3'. Because this is a bacteria system, obviously I already mentioned a cluster of genes forming a one transcription unit. Don't tell me you don't know. And this area we call it like Z, like Y, and like A. Okay. And what happened? Translation continue occur. Translation. What happened? Okay. So and I don't know. Seriously. And terminal, C terminal, also this one, different one. Okay. And terminal, C terminal. Okay, and C. Okay. So this lag-ZAG gene, right, will produce what? It's going to produce the beta-galactosidase. Beta-galactosidase. I think days, D-A-S-E-S. I forgot how to spell. And okay, what about lag-Y gene? Producing permease. Okay, let me check. Maybe I make some mistake with the MEASE. MEA MEASE, correct. Just the last one, need to remove it. Ah. Okay, MEASE. Okay, yeah. So, okay, this guy is not important. This is nothing to do with lactose metabolism. Okay, but permease has a role, not cutting the lactose, but is to transport the lactose into the cell, allowing, okay, once the permease bring the lactose into the cell, right, bacterial cell, because they can absorb inside, and then what happened? The beta-galactosidase will come and digest the lactose and cleave into galactose and glucose. Okay, but okay, this is what this is. Obviously, is the how they express when the surrounding full of lactose, right? Full of lactose, and then yes, the transcription will come and express. But what happens if it happens that no lactose at all? No lactose, what to do? Well, how are they gonna block this? How are they gonna block this expression? Block all this expression. How are they gonna block this expression? Well, the system tells us that, okay, the transcription will occur. How are they gonna regulate? So I just tell you that there's a lactose just now before this, right? I was telling you that it contains the lactose, and then the gene will transcribe into messenger RNA, and then later on will express, translate into protein, into enzymes, beta-galactosidase and permease, and then work in the lactose metabolism. Okay, but I did not tell you when there is no lactose. Okay, how are they going to stop this transcription and translation? Okay, now I'm going to tell you. So there is far away. There's a cute little gene. It's called LAC-I gene. Okay, LAC-I. Okay, far away. Okay, it's not part of the lag operon because it's really far away. Okay, so I put ... And what happened? Also, this lag gene will, let me erase the crosses. Okay, better. So the lag genes also will do what? Gene expression, transcription, and translation. And finally, it will get you a beautiful enzymes. Let me put a horse shoes, okay? Use, it's like a, ugly. Okay. So this lag gene will, well, it's a gene, will be transcribed and translated into a donut shape. I made it up, okay? It's producing a repressor. Okay, so that's a repressor coming, new kids on the block coming. What happened? So what happened? This guy will come and they will recognize a place. It's not called La La Land, it's called Lag O site. So the repressor will swim around and come and bind to the Lag O gene site. Okay, what happened? I know. So what happened is that they will come and bind to this site. So maybe I just move away this, erase these things. Too quick. Okay, it will come and bind to okay, it will bind to the Lag O genes, okay? But they bind to the Lag O genes. How are they going to stop? Well, basically, if you still remember, the Lag P is the promoter. First will be recognized by the sigma factor, right? And then the sigma factor actually is facilitating who to come and bind to the promoter site is the RNA polymerase. Okay, so I'm not going to draw the. So who is coming? So who is coming? Is this guy is who? So this guy is RNA polymerase. Okay, so the RNA polymerase bind to the promoter site because of the help of the sigma factor. So they have to pass through the Lag O gene and continue the transcription. Unfortunately, right now the repressor is binding to the Lag O gene, block the view. Okay, so the RNA polymerase stuck in the promoter operator site. They cannot move further. So therefore, gene transcription stop. No gene transcription. Okay, so no gene expression. No gene, no transcription stop. Obviously, you will not get any messenger RNA from this lag operon, right? And then therefore you don't get any beta galactosidase. Okay, so this one, I think the concept is very, very clear. Okay, so any question? This one is very important. So I want everyone to make sure understand because I'm going to continue. Actually, I didn't finish, but I want you to ask me question. Chuntet, Jialing, Jiayi, any question? Come on. Before I move to the next page. Very important, huh? No question? No question is okay, huh? No, I think no. Good. No question. Okay, so you understand. But right now, I'm asking you, hey, if there's a repressor there, right? Always the lag I gene express ma, it will not stop ma, right? So what to do if there's a repressor, if I have a lot of lactose in like high concentration, okay, go back to this this plate B. Okay, so now the plate A already explained to you that the no lactose, right? And then no gene expression, no expressions of the beta galactosidase, correct? Because there's a lag-I gene right here, lag-I gene producing the repressor. It's going to block the RNA polymerase to continue the transcription, right? So clear. But what about plate B? Plate B contain high concentrations of lactose, but we have a repressor continuously generated by the lag-I gene. So that's a repressor binding to the O site. But how am I going to express my beta galactosidase? It's not mine, it's bacterial one. So basically, when there's a lot of lactose, okay, let me do this. There's no more color that I can choose. Maybe green. Okay, maybe there's more color. I need some color that is really sharp. Maybe there's one. Okay, I put this green. Better save it. Okay, so here, wow. So lots of lactose. Lactose around here, lactose around here, lactose. So this lactose can actually recognize the repressor. Okay, and the lactose here will come and swim around and then come and bind to the repressor. So the repressor contain a site. What we call this site? We call this site is a ... It's the lactose binding site. Okay, so here, this is the lactose binding site. Lactose binding site. Okay, so there's a lactose binding site. So when there's a lot of lactose come, and then the lactose will come to recognize the repressor, not recognize, they will come to bind to the repressor, which is the specific binding site. And when they bind it, okay, this donut shape will open it. Okay, so no longer clapping on the repressor will no longer hold to the lag-O site. It will release from the O site, remove. Okay, remove. Okay, from the O site. It's a bit ugly. Let me just move away this. Okay. So once the remove away from the O side, so the whole thing will clear. There's no longer, okay, so this guy will no longer be there. Okay. What to do? My mistake. Undo. Okay, good. No. Okay, good. So you can see that the so this guy will no. Oops. So the whole thing, right? Lactose binding to the repressor, and the whole thing repressor will remove from the like O side. So the pathway is completely clear. Allow the RNA polymerase to pass through the O side and start to transcribe. Okay, transcription will occur, and then it will produce what? Produce the messenger RNA. So now transcription occur, and then so you can see that transcription occur. It will continue transcription and then translation, and then generating this beta-galactosidase and permease. Okay, so this is how the system works. Okay, here you are. And basically today I can't record because someone took the place. I came very early, so someone. I hope some of you please don't come too early to occupy my place because I came around 4:40, yeah? And then if you come to this classroom too early, and then you will have to do the recording for your friends. Okay, I can't just come too early because, yeah, 4:30 maybe, sometimes 4:30 I come, and then someone already used the come into the class, and then, yeah, so I cannot do any recording today. Okay. Okay, so I hope you understand. Okay, so this one already mentioned. So please remember that in this page, I mentioned that the permease, okay, is what is the functions of permease? Is to transport the lactose into the cell. Okay, once they transport the lactose into the cells, beta-galactosidase will cliff the lactose into glucose and galactose. Okay, glucose and galactose. So here, this is the system, and as I mentioned before. So when we talk about the the key things I want to talk about is that this lactose, right? Lactose bind to the repressor to induce the gene expression. We call this lactose as a inducer. English, I'm talking about English right now. So this is an inducer, can induce, okay, this gene expression, okay, or we call it induction. Okay. So this is all I already explained to you. Okay, the lac repressor, okay, encoder is a lac I gene, okay, encodes the repressor. You can say lac repressor, okay, block the expression, and promoter. Obviously, you already know. In bacteria, you have Tata box at -10 region, and another one, TTGACA, is what do you call that? Is at the -35 region. Okay. Obviously, in bacteria, sigma factor will recognize the promoter region and facilitating what? The RNA polymerase to bind to this site and to initiate the transcription. Okay. So lac O means lac operator. Okay. O means operator, and it's in between promoter and the lac-ZAC gene. Okay. It's essential for what? For the repressor to, that means the lac repressor, to bind to this site, to suppress or suppress the gene expression. Okay, as I mentioned, that the lac operon, including what? Including POZYA. Okay, but lac I gene is not part of the lac operon because they are really, really far away. Okay, so it's not part of the lac operon. And repressor can recognize here. This repressor has the DNA binding site that can recognize the operator. Okay, they have a DNA binding site that can bind to the operator and therefore bind it. And also they have the operator recognize the operator DNA sequence that bind lactose or analogs of lactose. Later on, we will talk about the analogs lactose. Okay, so the repressor will bind only to the sites of the DNA near the genes that is controlling and not to other sites distributed throughout the chromosome. So you can see that obviously this is the repressor. Okay, it comes and binds to the O site. Okay, suppress the gene expression. And the lactose here is a inducer, will come and bind to the specific site of the what do you call that, repressor. And therefore, once they bind it, so the whole system, the whole repressor will release from the O site and promote the gene expression. Okay. So here, this is all what I'm talking about today. And I hope you understand the whole system indeed. Okay, so we normally we do a lot of artificial expression, right? We don't use lactose. Okay, we use another inducer. We call it isopropyl beta dithiogallactoside. Here, this is the guy. Crazy name. So I rather you remember this. IPTG will be okay. IPTG. I means iso. P is propyl. T is thiogal. G is galactoside. Okay. So this guy will be able to be the inducer. Okay, can replace the lactose, and then we can add this IPTG. Why we want to add IPTG? Yeah, that's a lactose, right? Oh, because this IPTG cannot be cleaved by beta-galactosidase. Okay, remember that. So therefore, it is good for us to control the system, because at the end, this guy, IPTG, will not be cleaved by beta-galactosidase. We just need what? We just need to collect some gene-specific one that will not affected by the lactose cleaving by the beta-galactosidase. So using the IPTG is a good metaphor. Okay. So in the future, you're going to use the XGAL. Okay, so the XGAL is another type of diet. Okay, that you can how to say digest by beta-galactosidase. Okay, so here XGAL, this one. Okay, XGAL means 5-bromo, 4-chloro, 3-indole beta-D-galactopyronoside. Huge name. Okay, so you just remember this beta-gal. XGAL should be okay. XGAL. Okay, X-Man, XGAL. And this XGAL can be digested by beta-galactosidase. And forming the first thing is what? This guy, number 1, is called what? This is called 5-bromo, 4-chloro, 3-hydroxy-indole. But this structure is not stable. Okay, it will spontaneously dimerize. So it will double the structure. So it will form a double bond. Okay, and forming here like this. You can see that. So one is, so it will, bromo is here. So that's a bromo. Okay, so it will form 2 of this. Okay, will form the dimerization. It's oxidized into forming this indigo. When there's an indigo, means that it will turn blue color. Okay, it's a very good indicator. In the biotech industry, we use a lot of this technique. Okay, so please remember this pathway. Okay. Okay, so now we're going to talk something very serious. Okay, so lag origin. Well, when the lag origin, you have a mutant, right? Okay, what happened is that, let me do this. Maybe not. Maybe today. Maybe tomorrow we can talk more. We talk this. Okay, easier one. Okay, let's talk about mutant. Mutant Ninja Turtle, but this one is a mutant in bacteria. Okay, so lag I gene. When you have a mutant of this at this lag I gene, right? You can do like this. Okay, you remember this writing, huh? You have to remember. Okay, if this is a wild type, when we say wild type is a normal gene, okay, we say I plus. They are normal. Okay, so plus, good is plus. If there's a mutant completely mutated, okay, so we call it I minus. Okay, this is a simple form. There's more, okay, but I'm going to introduce today only this two, plus and minus. Okay, please remember. So when there's a plus, means this guy will generate a normal protein repressor. There's a repressor, functional repressor. What about this? This guy will produce a non-functional enzyme, totally, completely knockout, or completely not expressed at all. Okay, let's go for not expressing. So when you knock out this, when we say knockout, that means this gene, okay, let's make it simple for you guys. Okay, so this is a mutant, we call it I minus. So they're not going to express or produce any repressor, no repressor at all. Okay, so now you have a chromosome. If you still remember, the chromosome will pair and synapse, right? And then they are diploid stage, for example. So what happened? So you have a genes here. I just let you know that there's a maternal and paternal, right? For example, homologous chromosome. Just go for it. And so here, dot, 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 dot. And then here. So this is lag I plus, not good. Okay. Okay, so here you have one like origin dot 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 far away. So here you have P O Z Y and A. Okay, so here you have a I is plus. Okay, you have another gene. Okay, so let's say this is a maternal, this one is paternal, okay? I just made it up, okay? Okay, and then they are diploid. Okay, you have P, O, Z, Y, A. And what happened? This is I minus. So from this single cell, right, you have one I plus and one I minus. We do it like this, okay? Cross I minus, okay, which means that they are coming from a different chromosome. Okay, so this one and this one is in a different chromosome. So therefore you have a cross here. Sometimes people put 2 lines, okay? 2 line, and some people put 1 line. Okay? I want you to understand this. So what happened is that when you have, okay, so here, now I get you. So this one is I plus. It will generate a lot of repressor. Okay, repressor. And this guy is not going to generate any repressor, no repressor, no repressor. So this one is I minus. So therefore we call it I plus I minus. It's a heterozygotes, okay? So what happened? So you would think that, okay, so this genes, this is the chromosome, same chromosome from here to here, okay? So suppose this one I minus, right? It will affect the whole genes, whole chromosome here. Not really, okay? Because the repressor can swim around, okay? So they can swim around across the land, across the sea, and then they will come and bind to any operator as they wish. So they will bind to this operator and also this operator. So therefore, the gene will not be expressed because both operator, right? 2 chromosomes are all blocked by the repressor. The repressor are here bind to the operator site. So therefore, RNA polymerase cannot pass through this operator site to do the transcription. So there's no transcription occur. Okay? Okay. So that is all today. Okay. I want to talk about Hui Yan. Hello, Hui Yan.
Yes.
You came today
Yes.
very early.
Uh,
You came very
yes.
early. So can you please next time come to the classroom a bit late? can be sharp like
Okay.
5 o'clock lah, because you came too early, 20 minutes earlier. So I cannot do any recording today. Okay. So I'm not blaming you, but also other people also did the same last time. So always I have to. So please, boys and girls, okay. I'm not complaining anyone. I just hope that obviously